Hello, everyone, and welcome to today's webinar, Advancing Cancer Research with Tools for Liquid Biopsy. I'm Christy Jewell of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. Now, we encourage you all to participate today by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. Today's presentation is educational and thus offers free continuing education credits. You can find those continuing education credit buttons on the bottom of your screen. Just click on that button to obtain your credits. And with that, I'd like to now welcome today's speaker, Dr. Laure Jobert, Staff Scientist, Thermo Fisher Scientific, Norway. And also joining us today for the live Q&A are Dr. Iwona Grad and Dr. Ketil Peterson. For a complete biography on Dr. Jobert, please visit that presenter tab at the bottom right hand of your screen. Dr. Jobert, welcome. You may now begin your presentation.
Thank you, Christy. Hello, everyone. My name is Laure Jobert, and I'm a scientist at Thermo Fisher Scientific. I work in a team composed of more than 20 scientists and all dedicated to the development of magnetic bead-based assays. These assays aim at detecting and quantifying specific biomarkers from any type of sample matrix. And today, I'm going to present some tools enabling various analyses from liquid biopsy, all of which go to advance your research on cancer. I will first introduce the notion of liquid biopsy and why liquid biopsy is a precious technology in cancer research. Then I will go through different methods and workflows to isolate circulating tumor cells, CTCs, exosomes, and circulating tumor nucleic acid, ctNA. The presentation will end with a summary of the presented data. So what is liquid biopsy? According to the National Cancer Institute, a liquid biopsy is a laboratory test done on a sample of blood, urine, or other body fluids to look for cancer cells from a tumor, or to look for small pieces of DNA, RNA, or other molecules released by tumor cells into a person's body fluids. And liquid biopsy offers several advantages over tissue biopsy. First, it's minimal invasive and thus safer for the patient. Then it's also easier, faster, and thus cheaper to collect than tissue biopsy. And finally, in many cases, it provides a crucial molecular and response information in real time. So the most used and characterized body fluid in liquid biopsy is blood. So why can blood be used for cancer management? On the top left part of the slide is depicted a tumor mass. Cells from the tumor mass enter the bloodstream. These are circulating tumor cells. Most CTCs die shortly in circulation after 1 or 2 hours. However, a small fraction of CTCs will survive. Moreover, circulating tumor DNA, non-coding RNAs, vesicles, proteins, all depicted in red on the slide, also are secreted in the bloodstream, either passively by dying cells, or actively by secretion. This is still unknown. But what's important is that the analysis of all these elements present in blood has a tremendous clinical utility in cancer management, from early detection, primary tumor identification, treatment selection to monitoring. Monitoring includes therapeutic response, disease progression, and prognostic recurrence. Indeed, the three main tumor-derived elements present in liquid biopsy can be analyzed in various types of downstream, or can be used in various downstream analyses. This can be done for the detection and quantification of specific biomarkers or for the identification of genetic variations. All of this goes to enable prognosis, diagnosis, and monitoring. Let's start with the extracellular vesicles. Extracellular vesicles are found in the plasma and are mainly used for DNA and RNA sequencing and proteomic analysis. Circulating tumor DNA, also found in the plasma, can be analyzed for mutation or methylation profiling, copy number variations, and single nucleotide variants, either by PCR or by next-generation sequencing. Finally, the circulating tumor cells, CTCs, are found in the Buffy coat fraction of the blood, which, by the way, accounts for less than 1% of a whole blood sample. And CTCs are mainly used for cell enumeration and genomic analysis. So, liquid biopsy is a precious technology in cancer research and treatment. And in this webinar, we are going to address methods to enrich the main elements present in liquid biopsy, namely CTCs and exosomes. In a third section, we will address the DNA target enrichment technology prior to next-generation sequencing. So, 150 years ago, in 1869, the pathologist Thomas Ashworth provided evidence for the presence of circulating tumor cells in the blood of a metastatic cancer patient. He therefore hypothesized for the first time that circulating tumor cells are a fundamental prerequisite to metastasis. How does this work? Cells from the primary tumor acquire the ability to enter the blood circulatory system. Then CTCs move around and transit to distant organs. Finally, these cells can invade distant tissues where they divide and form a secondary colonization site. An interesting fact to remember is that the average patient with metastatic cancer has between 5 and 50 CTCs for every 7.5 milliliter of blood. So as said earlier in the introduction, CTCs are mainly used for cell enumeration and genetic or genomic analysis. But to do so, one needs to isolate the cells. On the top part of the slide is shown a workflow that allows the positive isolation of circulating tumor cells from whole blood. So what we have done here, we have used Dynabeads Dynabeads epithelial enriched that are coated with antibodies recognizing EpCAM. EpCAM is a surface marker on epithelial cells. And we have used these beads to isolate breast cancer cells that were initially spiked in the whole blood. Once the cells are isolated on the beads are washed and the isolated cells are lysed. Then the RNA is extracted and purified on Dynabeads oligo-DT beads. Then the CK19 mRNA is quantified, analyzed by reverse transcription and quantitative PCR. CK19 being another epithelial marker. The graph shows that the CK19 CT values obtained with the counted cells versus the CT values obtained with the cell-spiked blood overlap from 7 down to 1 spiked cell. indicating that Dynabeads epithelial-enriched beads nicely and successfully isolate the few breast cancer cells that were spiked in 7.5 ml of blood. However, there is a limitation to the EpCAM-mediated isolation, which is that it does not isolate all CTCs, because not all CTCs have EpCAM at their surface. Indeed, there is no universal CTC-specific antigen. So what we have done in our lab is that we have used Dynabeads coated with streptavidin, either C1 or T1 beads, in combination with a biotinylated anti-EpCAM antibody. We have used these beads to capture 5 breast cancer cells spiked in the blood of 4 different donors. The graph shows that Dynabeads streptavidin are as efficient as Dynabeads epithelial enrich to capture those cells. These findings also demonstrate that it is possible to isolate any CTC, even EpCAM negative, as long as you have a biotinylated antibody that recognizes a specific marker at the surface of your cells of interest. So these were our internal data. But there is also this interesting study from Wu et al. Published in 2020, and the authors have looked for enrichment ways of rare tumor cells from PBMCs. Basically, they have pre-labeled cells from 3 different human pancreatic cancer cell lines, and they have spiked them in PBMCs concentrated in 1 ml in order to mimic diagnostic leukapheresis samples. Several types of Dynabeads bound to anti-EpCAM antibodies were used to capture the spiked cells. The whole workflow was automated on the Kingfisher Duoprime instrument, and the recovery rates shown on the graphs were calculated by microscopy analysis. You can see on the graphs that the highest recovery rates were obtained with either Dynabeads epithelial enrich, shown with the blue dots, for the first and third cancer cell lines used, or with the Dynabeads biotin binder, as shown by the red dots for the CAPAN1 cells. These recovery rates were in line or even slightly higher than the ones obtained with the cell search system, as shown in some supplementary information from the same publication. And for those who don't know, the cell search system is the first and only FDA-approved system to detect and characterize CTCs. There was no statistically significant difference in the recovery rates when medium or maximal amounts of beads were used. And it's also interesting to note that the recovery rates were highly dependent on the cancer cell line that was used as a source of rare tumor cells, illustrating the heterogeneity in the surface markers of the cancer cells, on the cancer cells. In the same study, the authors have also looked for alternative strategies to enrich CTCs. And to do so, they have used an additional marker, MUC1, tested in the Kingfisher Duoprime for the capture of a pre-labeled CAPAN1 cell spiked into PBMCs. The graph on the panel number A indicates that when MUC1 alone was used, the recovery rate dropped compared to EpCAM, looking at the light blue dots compared to the gray ones. When MUC1 bound dynabeads and EpCAM-bound dynabeads were combined in the same enrichment step, only partial increase of the recovery was achieved, as shown by the dark blue dots. Yet more interestingly, when a sequential enrichment was performed, either with MUC first or an EpCAM second or the other way around, the recovery rates obtained were similar to the ones obtained with EpCAM alone. And even more interesting, the sequential enrichment led to the capture of two separate populations, as shown on panel B. So these findings demonstrate once again that it is possible to isolate CTCs with any antibody that is biotinylated in combination with beads coated with streptavidin. It's also interesting to see that a sequential enrichment might help characterize subpopulations of cells. So besides or contrary to the positive isolation of CTCs, it's also possible to enrich CTCs by a negative isolation. This can be done with dynabeads coated with CD45, CD45 being a specific marker of leukocytes. So the beads coated with CD45, antibody against CD45, will isolate the leukocytes, indicating that the supernatant will be enriched for target cells. And the analysis of the supernatant shows that more than 99.9% of the leukocytes were depleted. The untouched enriched target cells can then be analyzed further by imaging, an NGS, PCR, or can be used for further targeted positive isolation. So altogether, these data have shown the powerful use of dynabeads for CTC enrichment, either by positive isolation using dynabeads epithelial enrich, or using dynabeads streptavidin in combination with biotinylated antibodies specific for CTC markers, or by negative isolation using dynabeads CD45 or dynabeads CD15. The latter increases granulocyte depletion. So, as previously mentioned in the introduction, another key tumor-derived element present in liquid biopsy is exosome. So now we are going to review methods to enrich exosomes. What are exosomes? Exosomes are a heterogeneous group of small vesicles ranging from 30 to 150 nanometer in size. They are secreted by all cells, including cancer cells, and found in most body fluids. Exosomes play an important role in cell-to-cell communication. They can cross the blood-brain barrier, making them potential drug delivery system in brain cancer. An interesting fact to remember, again, is that exosomes are the most abundant analyte within the liquid biopsy, reaching 10 to the power of 11 particles per ml of blood. And in a cancer patient, up to 10% of the total circulating exosomes are tumor-derived exosomes, depending on the tumor stage. So, the traditional isolation methods of exosome mainly include differential ultracentrifugation and size exclusion chromatography. But these methods are cumbersome and complex with challenging and time-consuming operations. So today, I'm going to review two other methods, bead-based isolation and polymer-based precipitation, that are easier to carry out and give higher yields. Let's start with the bead-based generic enrichment of exosome. This method is based on a physical property of exosomes. These are negatively charged particles in a hydrophilic buffer. That means that they can be captured on a positively charged beads, acting as an anion exchange, a strong anion exchange solid phase. This process is reversible by ion exchange. So this enables the release of exosomes if that is desired. The whole workflow can be easily automated on Kingfisher instruments, and the downstream analyses that are compatible with this enrich release method are numerous. Here are examples of downstream analyses that are compatible with this method: transmission electron microscopy, particle analysis, flow cytometry, Western blot, and mass spectrometry. In the next slide, I will look deeper into particle analysis with the use of the Spectrodyne instruments. So here, what we have done, we have collected exosomes from SW40 cells medium. Once we have them, we have enriched and released the exosomes by ion exchange using the Dynabeads intact virus enrichment beads. Then the exosomes were further analyzed for size, concentration, and phenotype by microfluidic resistive pulse sensing. Resistive pulse sensing is the particle size analysis method that is used worldwide to quantify red and white blood cells in a whole blood analysis, and this method is also very well known as Coulter counting. But the difference here is that the analysis chamber is replaced by a microfluidic cartridge, thus enabling the analysis of particles that are much smaller. So the size distribution and concentration of the exosomes was assessed on the NCS1 instrument that is shown on the bottom left of the slide. Two different types of cassettes were used: C400 for particles between 65 and 400 nanometer, and C2000 for the analysis of particles between 250 and 2000 nanometer. The two top graphs show that the majority of the particles present in the sample had a size below 100 nanometer. Moreover, the concentration of the input sample was estimated to be around 10 to the power of 10 particles per ml. Moreover, the phenotype analysis was performed on the ARC instrument, the second from the left, shown on the slide. And this phenotype analysis confirmed the presence of CD81 positive exosomes in the isolated fraction, as shown by the blue curve on the bottom right graph. So we have seen now the generic enrichment of exosomes by ion exchange, but it's also possible to capture or to isolate and detect exosomes using specific antibodies. The transmembrane markers from the tetraspanin family of proteins are well-characterized exosomal markers, among them CD81, CD63, and CD9. On the left part of the slide, the exosome human CD81 isolation reagent was used to capture exosomes prior to analysis by Western blot using another CD81 antibody. On the right part of the slide, the exosome human CD81 flow detection reagent was used for exosomes capture prior to detection by flow cytometry. So these two products or two reagents were developed in such a way as to help maximize either the isolation efficiency or the detection by flow. So the generic enrichment of exosomes by ion exchange, followed by release, and the specific detection by Western blot or by flow cytometry can be successfully combined for various types of liquid biopsy. And this is shown here with serum. So here, both enrichment and release steps are shown to be very efficient by Western blot using an anti-CD81 antibody. The intensity of the signal is inversely proportional to the serum concentration, which might be due to some inhibitory effects coming from the serum. On the right part of the slide, both enrichment, release, and detection steps were shown to be efficient from serum. Note the presence of endogenous exosomes in the unspiked sample, as shown by the blue peaks. Urine samples were also tested in a very similar experimental setup, except that exosomes were spiked in urine samples. And again, both Western blot analysis and flow cytometry analysis confirmed the efficiency of the enrichment, release, and detection of exosomes from serum. Here, two different markers were used, CD9 and CD81. Finally, we will go through a last method to isolate exosomes, which is a bead-free method and is named polymer-based precipitation. As already said in the introduction, this approach, together with the bead-based methods, is faster and gives higher yield compared to the standard ultracentrifugation procedure. The principle of the method is that the reagent, the precipitation reagent, ties up water molecules and forces less soluble components, such as exosomes, out of solution. This allows the exosomes to be collected after a brief and slow centrifugation. So in this study, published in 2013, and actually one author of this publication is in the room, Katia. And what has been done is that exosomes from 3 different serum samples were isolated using either ultracentrifugation or the precipitation reagent. The exosomal marker CD63 was analyzed by Western blot, and that showed that the signal, the intensity of the signal was slightly higher when the precipitation reagent was used. Moreover, the levels of 5 microRNAs and 2 messenger RNAs that were previously reported to be present in exosomes have been analyzed by reverse transcription and quantitative PCR. And the graph in the middle shows that the precipitation reagent led to similar or slightly higher levels of RNA compared to ultracentrifugation. At the right part of the slide, a TEM picture is showing that this precipitation reagent can isolate exosomes to be then analyzed for phenotype and morphology. Here, the isolated exosome is immunolabeled with anti-CD81 antibodies, followed by 10 nanometer protein A gold nanoparticles. So here is a summary of the different methods we have reviewed for the enrichment, release, and detection of exosomes from liquid biopsy. We have seen the specific capture using specific antibodies recognizing exosomal markers. We have seen the generic capture either by ion exchange or by precipitation, and we have looked at detection using, again, specific antibodies. So in some of my previous slides, I have mentioned a few times Kingfisher instruments for the automation of the workflow. All the methods we have seen so far can be, of course, be done manually, but can also be automated to save a lot of hands-on time. The Kingfisher duoprime instrument is ideal when a limited number of samples is processed. For medium throughput, the Flex and Apex instruments are best suited. The advantages of Apex are that this instrument can not only heat the sample, but can also cool the sample. It has a UV mode, it has a barcode reading, it's connected to the cloud. That enables the automatic download of the scripts. And it also has 2 magnet heads and blocks that can be very easily and quickly swapped, depending on the number of samples. And finally, for high throughput analysis, the Kingfisher Presto is used in combination with liquid handlers, such as Hamilton or Tecan instruments. So in the last section of my talk, we will focus on the genomic readout from liquid biopsy. Liquid biopsy is mainly used for genomic analysis. And as we have already seen it in the introduction, liquid biopsy contains many different types of nucleic acid. Circulating tumor DNA, circulating tumor RNA, nucleic acid from CTCs, nucleic acid from exosomes, and all these elements can be subjected to analysis either by PCR or by NGS. PCR enables a fast turnover, but is limited to selected targets. It's cost efficient, and the relatively new technology of droplet digital PCR enables very high sensitivity, much greater than classical qPCR. NGS is high throughput technology, providing a comprehensive and unbiased analysis. It's also cost efficient if multiplexed. The specificity of tumor nucleic acid in liquid biopsy is that it's present in low abundance. That means that a target enrichment step might be required. And one way to do target enrichment is by hybridization capture during the library prep step. And once again, magnetic beads are a powerful and very useful ideal tool for this purpose. So we have compared our Dynabeads streptavidin for target enrichment product with other beads available on the market for the library prep yield. Library prep yield is a critical parameter for a successful sequencing analysis. The graph shows that Dynabeads enabled the highest library prep yield compared to the other tested beads, with a concentration similar to the control sample. Another key parameter or critical parameter for successful NGS is coverage. Coverage describes the average number of reads that aligns with a known reference basis. For example, a 30x coverage will have an average of 30 reads spanning. In that case, you can see on the graph that all tested beads had nearly 100% coverage. However, when a deeper coverage is required, and that's the case for clinical application or for the identification of rare variants, in that case, Dynabeads performed better than the other tested beads. So we have just shown that a robust and efficient enrichment of very fast and good enrichment of target sequences can be achieved for the preparation of NGS libraries using Dynabeads streptavidin for target enrichment. So to summarize, we have seen that liquid biopsy that are collected from patients, they contain precious elements such as vesicles, cells, nucleic acid, proteins, all of which can be isolated on Dynabeads magnetic beads for further analysis. So Dynabeads are an ideal tool to study cancer. So today we have reviewed methods and workflows for the isolation of cells and exosomes, as shown in the middle of the slide, and also methods for sequence enrichment prior to NGS. But I would like to mention that we have other products used in other workflows for liquid biopsy analysis, which are the Dynabeads magnetic beads for protein isolation, for subsequent proteomic analysis. And we also have Dynabeads and MagMax kits for nucleic acid isolation for subsequent NGS. I also encourage you to go on our website, where you will find useful information in the form of brochures, application notes, or poster. And here are links to some of them. And finally, I would like to, before we open the floor for questions, I would like to introduce two colleagues of mine who will join me for the Q&A session, Dr. Ivona Grad and Dr. Ketil Pedersen. Thank you for your attention.
Thank you, Dr. Jobert, and welcome, Dr. Grad and Dr. Pedersen. We will now start our live Q&A portion of our webinar. To our audience, if you have any questions you would like to ask, please submit them now. Again, just click on the Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. So, Dr. Jobert, over to you for the Q&A.
Thank you, Christy. So we have one first question that I can take. Is it possible to release CTCs after capture on beads? So if the release of CTCs is required for the downstream application, yes, it's possible. We would then recommend the selection product, the selection epithelial enriched Dynabeads. These beads enable the isolation and the release of viable cells. And the principle is that there is a DNase 1 treatment or present in the release buffer that will cleave a linker, a DNA linker between the bead and the EpCAM antibody. Just have to be aware that then the EpCAM antibody will remain bound to the enriched cells, but that's possible. Maybe, Kjetil, you take the next question.
Yep.
You have to read it.
Yeah, that's the thing.
Okay, I can read the question.
Please do. Thank you.
So question number 2. You have shown data on the generic capture of exosomes on beads, but the name of the bead product is Dynabeads Intact Virus Enrichment, not exosome. Why?
Yeah, thanks a lot. Yeah, that's a very good question. And this product was actually developed during the pandemic, the SARS-CoV-2 pandemic, the COVID pandemic, as a response to that pandemic. So we started to develop this product to capture SARS-CoV-2 virus for several downstream applications. But we knew that this product, as it's a strong anion exchange bead, would also capture exosomes. And we actually used, during development of the product for SARS-CoV-2, we used exosomes also as a model vesicle. So that's the reason why we still have maintained the name of the product as a virus enrichment bead, but it works, as we have seen now, very well for capturing exosomes as well. But we have also tested it for other viruses. I would like to mention that. A range of different viruses, both from captured from cell culture media, virus transport media, but also wastewater samples. So many different viruses this bead will work to capture. Yeah. Thank you.
Okay, let's take the next question. The next question is, which anion can be used for exosome release after bead-based generic capture?
Yeah, so this is quite simple actually. It's based upon the affinity, the anion affinity, which will, or an anion with a higher affinity will outcompete the bound exosomes or even the bound virus. So we have tested several different anions, like potassium iodide, high concentrations of sodium chloride, but also citrate ions, which works well.
Okay. Then there is another question on the Dynabeads streptavidin. Are there any chemically differences between Dynabeads My1 streptavidin T1 and Dynabeads streptavidin for target enrichment? Shall I answer?
Yeah.
So we cannot, I think, really provide more information on what are exactly the chemical or the functionality of Dynabeads streptavidin for target enrichment. But what I can say is that these beads have been optimized for this specific application. So we know that the functionality of these beads is made in such a way to have a good capture of DNA sequences during library prep. I don't know if you want to add anything. Okay. There is another question about CTCs. So the question is, does the cell release using selection kit damages surface markers of the CTCs? To my knowledge, no. I don't know if you have any more experience.
No, also to my knowledge, that's not the case. No.
I mean, the principle of the release for this product is the cleavage by DNase 1 of this DNA linker, as I said before, between the bead and the EpCAM antibody. So, and there is no reason how this release buffer could damage any protein at the bead surface, the surface of the cell, sorry. Okay. I don't know if there are any other questions.
It looks like those are all of our questions. Do all three of you, any of you, have any final comments for our audience?
Thank you a lot for your attention, and we remain available for any other questions you might have via email or other way. Thank you.
Very good. Thank you, three, for joining us today. And thank you for your presentation and for your important research. We also want to thank Labroots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. But before we go, I do want to thank the audience for joining us today and for their interesting questions. Any questions that are submitted during the on-demand period, they will be addressed by our speaker via the contact information you provided at the time of registration. Today's webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thanks for joining us, everyone. Until next time, bye-bye.